Thiencarbazone is an active ingredient in multiple herbicides. The active ingredient is the component of the herbicide that kills or suppresses the plant. Herbicides are chemicals farmers use to kill unwanted weeds and plants in their fields. In the agronomy world, when a farmer grows a crop, unwanted weeds grow with it. Those weeds need to be killed or suppressed so that they aren't taking valuable nutrients from the crop, and the crop can then yield higher, bringing in higher profits.
There are many different types of weeds and therefore many different types of herbicides. There are two main categories, selective and non-selective. Thiencarbazone is a selective herbicide, meaning it only kills specific weeds. All herbicides are categorized into groups based on their mode and site of action in killing a weed at the molecular level. Thiencarbazone is a group 2 herbicide, which is a group that attacks the acetolactate synthase enzyme.
The acetolactate synthase enzyme is the first step in the synthesizing of branched-chain amino acids. The ALS enzyme uses two molecules of pyruvate to create the acetolactate molecule and carbon dioxide. At completion, the branched-chain amino acids valine, leucine, and isoleucine will be synthesized. These are 3 of the 9 essential amino acids and can't be synthesized elsewhere. When these amino acids aren't synthesized, the plant's metabolic processes start to shut down and the plant dies.
Symptoms that are synonymous with thiencarbazone are stunted growth, leaf chlorosis, interveinal chlorosis, leaf purpling, and then potentially necrosis and eventual death. These symptoms will typically show in 7 to 10 days, with weed seedlings being affected faster and larger weeds taking longer to show symptoms. The bigger the weed is, the greater chance there is for the weed to just be suppressed rather than killed.
This is the acetolactate synthase enzyme. There are multiple binding sites on this enzyme, depending on what group 2 herbicide is applied. For thiencarbazone, the main binding site is highlighted here. The molecule binds to 4 residues at this site. The thiencarbazone molecule is highlighted red, and the residues it binds to: serine 653 highlighted in green, arginine 377 highlighted in light blue, methionine 570 highlighted in purple, and tryptophan 574 highlighted in orange.
Other research conducted has shown that there might also be another binding site in which thiencarbazone interacts with. That is shown here with there being 5 residues at this site. So in all, thiencarbazone could potentially interact with about 9 or 10 residues in the acetolactate synthase enzyme to shut down the plant's metabolic processes, in turn achieving the goal of controlling the weed pressure in a farmer's field.